It just takes a second. Let me just like hit the chat so we can get rid of this. It's just been sitting there.
Oh, okay.
We good? Please remember to repeat questions that are good. Okay. So now we have Vito Marino from the Cohelis Lab. Did you want me to tell your title? I forgot to ask you. Or do you want to say it?
You can say it.
Okay.
Makes it less on me.
So he will be telling us about the development of an ex vivo intestinal organoid model to study epithelial changes in menopause. He also did sports. He did baseball. But what he really wants you to know is that he has 3 tattoos. And one
Why
of them-
more than that? But these 3 are the most important.
So one of them is of the heart, one of them is of the brain, and one of them is of the lung. So thank you, Vito. You have the floor.
I'm still in the gut.
No, I thought about it. I like to be symmetrical. So I have the lung here, so I need the gut here. That's right.
I'm the ovaries.
And the ovaries. All right. Thank you, Sarah, for that introduction. So menopause is a condition that most women will experience during their lifetime. A statistic is that about 6,000 to 7,000 women per day will enter into menopause. And this is a staggering statistic. And since due to the fact of increased life expectancy, women can expect to live about 40% of their lives in menopause. During the reproductive years, women more often than not will have consistent cycling that will result in homeostatic levels of 17-beta estradiol. Once women reach the perimenopausal period, their cycling begins to become inconsistent, which results in fluctuations of their 17-beta estradiol levels. Once they reach menopause, their cycling will cease, and therefore their 17-beta estradiol levels will plummet, therefore resulting in low circulating estrogen levels within the blood. In the last 2 decades, more women than men have died from cardiovascular disease. And also, it's been seen that women are less likely to survive their first heart attack compared to men. In this study, females and males of various ages that were diagnosed with heart failure were followed up for 12 years, and their progression to class 3 or class 4 or heart failure or stroke death was monitored. As we can see that females overall are worse off in their progression of heart failure compared to males. However, females above the age of 50, which is around the average age that women will enter into menopause, have a much worse progression of heart failure or heart failure death compared to females that are younger than 50 years of age. Another thing is that menopausal females are more predominant to severe symptoms of irritable bowel syndrome compared to premenopausal women. And irritable bowel syndrome is highly linked to a phenomenon known as leaky gut syndrome. In this study, this group decided to measure epithelial resistance of a colonic cell line at different time points using different concentrations of 17-beta estradiol. As it can be shown here that in a dose-dependent manner, 17-beta estradiol levels were able to increase epithelial resistance. However, when this group decided to treat with ICI, which is a non-specific receptor antagonist to estrogen, we see that the effects of estrogen to increase epithelial resistance is lost. This suggests that 17-beta estradiol is able to modulate gut permeability and epithelial resistance. So our lab is interested in showing how these all are somehow connected together and how the effects of loss of estrogen during menopause is able to affect the gut that is able to modulate cardiovascular health. So our lab had a general hypothesis that menopausal mice are more susceptible to heart attack. So we decided to take pre, peri, and menopausal mice, and they went under ischemia reperfusion, where hearts post-sacrifice were stained with TTC and Evans balloon. We were able to measure infarct size by taking the area of necrosis outlined in white and the area at risk outlined in black, multiplied by 100 to give us the percent infarct size. As you can see to the graphs to the right, that menopausal mice had a significantly higher percent infarct size compared to premenopausal mice. However, perimenopausal mice had no significant difference compared to premenopausal mice, which suggests that even though during peri their cycling is irregular, they're still able to have a sustainable amount of estrogen to be cardioprotective. So now we were able to figure out a piece of this puzzle that in menopause, there's an increase in infarct size and increased pathological effects due to cardiovascular events. Our lab also was very interested in seeing how does the gut modulate heart health. So in this study, patients who were diagnosed with atrial fibrillation had serum lipopolysaccharide or LPS levels measured. And over the time of 150 months, they measured how many of these patients survived over that time. LPS is known as a product released by microbes that are able to induce an inflammatory response. As the data shows that individuals that had relatively very high levels of LPS had much more rates of death due to atrial fibrillation and major adverse effects to cardiovascular events compared to individuals that had lower LPS levels.
And this is LPS in the blood.
Yes, it's serum LPS. So our lab was interested in looking at how the probiotic Bifidobacterium animalis subspecies lactis 420 or B420 was able to mitigate infarct size in mice. So mice that were treated with normal fat and high fat diet, and whether given a saline treatment, B420, or treatment of Lactobacillus salivarius or LS33, had an effect on infarct size post ischemia reperfusion. So using the same idea of taking the area of necrosis divided by the area at risk, we were able to measure infarct size. And as we can see in both cases of the diets, that treatment with B420 was able to significantly reduce infarct size compared to saline and LS33 treatment. This shows that we're able to target the gut to be able to mitigate these effects against major cardiovascular outcomes. However, the effects of B420 are cardioprotective specific, as other strains such as LS33, which is a commercially sold probiotic used in patients that have inflammatory bowel diseases, was not able to mitigate these effects. So the hypothesized mechanism that B420 is able to possibly mediate the protection against myocardial infarction is that ingested B420 can able to make resins within the gut or undergo processes of fermentation that will activate T regulatory cells. T regulatory cells, when activated, can either decrease the presence of pro-inflammatory cytokines that are known to exasperate myocardial infarction, or the activation of M2 macrophages that are able to have a reparative effect, mitigating the effects of a myocardial infarct. So our lab was able to figure out that the gut microbiome is a possible target used as a treatment to decrease cardiovascular events and the pathological changes that may occur. So our lab wanted to now see what is the effects of the loss of estrogen in menopause have on the gut microbiome. In our lab, premenopausal and menopausal mice that were control treated or treated with oligofructose or OFS, which is an inulin-like fructan, or also a prebiotic to feed the gut, had cecum samples taken and their gut microbiome was sequenced. At the genus level, we see that with OFS treatment, there's an increase in the genera of Bifidobacterium, which we have seen in our lab to be cardioprotective. However, in menopausal mice, we see that the genera of Allostepes is drastically reduced. And Allostepes is an important species known to release the enzyme beta-glucuronidase. Beta-glucuronidase is an enzyme able to deconjugate compounds that are released in the bile from the liver. They remove glucuronic acid, which is also known to be added onto estrogens that are released in the feces. There's over 300 unique genes of beta-glucuronidase, where 10% of them are able to deconjugate estrogens. And half of that 10% is able to deconjugate 17-beta-estradiol, which is the active form that is present within premenopausal females.
Is the proposition that it's being taken up into the blood with beta-glucuronidase?
Yes. So the beta-glucuronidase in the gut is able to deconjugate the estrogens that are being released in the bile, and that gives a chance for that estrogen to be recirculated back into the blood. Actually, there's a, I forgot the exact number, but there's a high percent.
I don't mean to stop.
Oh, no, you're
I'm trying to
fine.
stay on it. That's all.
So now we know that in menopause, there is a shift in the gut microbiome that may mainly be affecting these species that express these 17 beta-glucuronidase enzymes. Now our lab wanted to see how do all of these tie together, and we hypothesized that menopausal mice that are given a pre or probiotic are able to mitigate cardiovascular events. So in this study, menopausal mice were treated with B420 or saline, and they underwent ischemia reperfusion and infarct size was measured. We see on the graph to the right that females that were treated with B420 have significantly reduced infarct size compared to saline-treated menopausal females. Also in our lab, thanks to work done by Christina, that menopausal mice that were treated with oligofructose had no significant difference in infarct size compared to premenopausal mice, whether they were OFS treated or control treated. As the pictures shown to the right as a qualitative look, in panel B is the heart of a menopausal mouse that was control treated undergoing a permanent ligation myocardial infarct. As you can see in the white tissue as necrotic parts that has the infarct, and in panel C, you see a menopausal mouse post-MI that was treated with OFS. So our lab was able to now see that menopause and a gut treatment using a pre or probiotic was able to decrease infarct size and decrease the pathological outcomes of cardiovascular events. But however, our lab is not exactly sure about the possible mechanisms that mediate the loss of estrogen connecting to the gut and how that mediates inflammatory responses that result in exacerbating these myocardial events. So in the case of a premenopausal mouse, estrogen that's whether circulating in the blood or estrogen that was conjugated by the liver and released in the bile that's flowing through the lumen. So from the case of the lumen, increased beta-glucuronidase activity is able to have reabsorbed estrogen back into the body. And this level of homeostatic estrogen in the circulation is also able to enter the gut. Estrogen here is able to affect the enterocytes, increasing tight junctions and decreasing permeability. Estrogen is also able to modulate T cell differentiation. And both of these effects, due to a decrease in the translocation or endotoxemia due to the decrease in permeability and decreased T cell differentiation, we will have less T1 and T17 helper cells that are releasing pro-inflammatory cytokine release. However, in menopause, estrogen already that's low in the body, an increase in release of estrogen from feces would have a decrease in estrogen present in the gut. This will allow for a decrease of tight junction formation and an increase of permeability and a suppression of the inhibition for T cell differentiation. So therefore, the increased bacterial translocation or endotoxemia is allowed to be able to activate T cells, which result in increased T1 and T17 helper cell differentiation and increased pro-inflammatory cytokine release that results in increase in circulating cytokines, resulting in a low-grade systemic inflammation that we may believe is exacerbating myocardial events. Although I present this, our lab is still not sure whether or not this is an inside-out or an outside-in mechanism. We either believe that menopause first results in a shift of the gut microbiome that results in metabolites released by that shift in the gut microbiome that's resulting in these changes in the epithelium that's resulting in this pro-inflammatory cytokine release, or if it's an inside-out mechanism where the already decreased circulating estrogen within the blood is affecting the epithelial layer that results in bacterial translocation, therefore increasing pro-inflammatory cytokine release. So my project goals is to develop an ex vivo model using intestinal organoids from pre, peri, and menopausal mice to study the mechanistic changes in intestinal epithelium during the menopausal transition. And we also plan to characterize the transcriptome of ileal epithelium and immune cells resident to the lamina propria during the menopausal transition. So the methods and future directions that we plan to do for this is that pre, peri, and menopausal mice that would be treated with high-fat diet 1 week prior to sacrifice, which is an optimal amount of time to allow for shift in the gut microbiome and allow for any genetic and morphological changes that can occur within the gut. We will remove the ileums and we can isolate crypts to be able to create organoid cultures. These organoid cultures from pre, peri, and menopausal mice can undergo RNA sequencing, where we are able to generate hypotheses about the difference in transcripts between the groups. This also can be helpful as to validate our model that using in vivo guts to create this culture is able to have differences to study the particular mechanisms that we are interested in. The organoids can also be plated in a transwell assay where we can treat with stressors such as LPS or flagellin in the presence or absence of estrogen. Being able to manipulate the media of the basolateral or apical sides will be able to help us identify whether this is inside out or outside in mechanism related, seeing if changes of one versus the other stimulates a similar thing that we see in vivo. The ileum can also be taken and single cell isolates can be created from the ileal epithelium as well as a lamina propria to get possible resident immune cells that are present within the tissue. These isolates can undergo single cell RNA sequencing where we are able to also generate hypotheses about the differences in transcriptome between the groups based on hormonal status. We are also able to identify different cell populations that may be present within the differences between the groups that is across the menopausal transition. So far, the work in my project has been optimizing the crypt isolation and also the single cell isolation of the ileal epithelium and the lamina propria. So the image to the left is an image of isolated crypts that are highlighted in the red boxes, which you can see that we are able to successfully remove crypts from ileal tissue, and these can now be used for creating organoid cultures. And then to the right, as a picture taken from a microscope on a hemocytometer, is our successful protocol in removing single cell isolates of the epithelium that we are able to pull together to be sent for single cell sequencing. And with that, I would like to acknowledge my PI, John, in the Canhelis lab, especially Christina, for helping me with a lot of the experimental design and also just to bounce ideas off of. I also would like to thank the Zabros lab and Gialdi for helping us with the organoid protocol and the organoid cultures. I also like to thank the Churko lab and Ben for helping us with the single cell sequencing and all the protocols necessary for that. Also like to thank Frank and the Duca lab for helping us with the sequencing of the gut microbiome data. Also, thank you to the Granzer lab and Josh for doing a majority of our MI surgeries at the time. And also thank you to Physiological Sciences, and also thank you to Kate, who's sadly no longer with us, but she was a very big help during when I was entering the program. And also thank you to our funding from the Starboard Heart Center and the National Institutes of Health. And also thank you to BioRender, as that's where a majority of my figures were made. And I will take any questions.
Fascinating talk.
Thank you.
So I got a lot of questions, because I'm interested, obviously.
I'm hoping the dogs kind of like.
It's a dog?
Yeah.
That's not too dog.
Oh, those are my two babies.
So, you know, you talk a lot about the epithelial changing in many
Mhm.
words. You have that beautiful schematic.
Mhm.
My question is that hypothesis driven, or is that been shown by others that the gut is leakier? Because you then show this very elegant experimental design, but have you actually measured with a Lucene chamber, or, you know, taken the gut out of your animals and shown that they are leakier? Does the transepithelial resistance has changed?
Yes, so there's data that we have from our lab that wasn't in this presentation, but it's done that from in vivo doing a dextrin assay that menopausal mice have a increased leaky gut compared to premenopausal mice.
Okay, that might be something you want to put in your intro.
Yeah, and that data is still being analyzed, but we haven't analyzed it, we just haven't put it into a complete figure yet.
Okay, and then the other question, that cardiovascular data you showed was dramatically different, right?
Mhm.
The probiotics reducing the intoxiciants,
Mhm.
right? So my question was, first, did you give the probiotic before the event?
Yes.
Okay, and was there a change in circulating T regulatory cells in that model?
Yeah, so In the paper done by our lab, it was shown that in that study that treating with B420 increased the activation of T regulatory cells.
You said there wasn't published.
No, so this paper, so this mechanism was published in the paper that was with this data. So this was a paper that was released from our lab in 2017.
But did you actually measure the T regulatory cells in those animals? Are they?
Yes.
Are they increased?
No.
That was the important question, is that we were surprised not to see that increase. So we went and then measured their supposed activity through, distillation, also through activation. Yeah, so that was a big surprise. It wasn't a small amount.
Is there,
Dr. Duca has a question.
oh, do you
His
mind
hand is
reading it?
raised.
Oh,
So just say, Dr.
All right.
Duca, go ahead.
Dr. Duca, that's your
Can,
question.
can you hear me? Good.
Yes.
Cool. Awesome. Okay, great talk. So I guess my questions are a little bit on the organoid system.
Mhm.
So the first question is, you're going to take cells from premenopausal, perimenopausal, and postmenopausal,
Yes.
and then develop organoids. So the first question is, do you think that they're going to go back into like, I don't know, like a normal early state as you grow them out? You know what I'm saying? It's like you're taking a taking a menopausal organoid and a perimenopausal organoid, but you're taking it from the crypts. So do you think they're going to kind of go back to their original formation as they were in like a premenopausal? And then my second question would be, you're looking at the organoids, but your model is highly incorporating the gut microbiome. But when you use an organoid model, that's literally the first thing you're eliminating, is you're taking out the microbiome. So how do you think you can work around that or address that fact that you think you have a microbiome based method, but you're using a model that does not incorporate the microbiome?
Those are great questions. So to address your first question, talking about if the pre-peri or menopausal mice will go to a predisposed state where those changes may be lost. So that's kind of the point of doing the organoids and doing the RNA sequencing, is to see if the crypts are able to maintain those changes that are seen in vivo into a cultured model. So that's kind of the first step of creating the organoid culture is to validate the model that the changes are maintained once it's out of the body. However, due to some papers that I've read, estrogen is able to modulate the epigenetic changes within crypts of the gut. So I think that maybe possible loss of estrogen is able to change these epigenetic changes of the gut. So there actually might be maintained crypt changes that we might see in culture once we get the organoids. And then to address your second question about the gut microbiome. So yes, it is very gut microbiome heavy, and we plan to take out that part once we have the organoids. However, we're able to try our best to simulate situations that involve the gut microbiome, whether that be due to stressors such as LPS or flagellin. And we also have thought about doing conditioned media experiments where we could take strains of probiotics or different bacterial cultures that would be present in the gut microbiome and have the conditioned media to be put on top of the organoids to see how do they address that kind of environment.
So just to clarify, are these 2D or 3D?
So we can do both 3D and 2D. For the experiments that we plan to do with the gut microbiome related conditioned media would be in the 2D, as we would want to distinguish between a basolateral and apical side of the enterocytes.
So, okay. If that's the case, then I would suggest maybe doing a 3D, because what you're saying with a 2D, it's going to be harder to place the microbiome in those areas. But if you have a 3D, you can inject the microbiome into the organoid in the middle, because a 3D intestinal organoid is almost like a sphere in a sense. So you can inject it into the inside, which would be the apical side, and you know it's in there. You can micro inject it. Versus if it's 2D, right? It's going to be tough to put it inside. It's going to be a lot tougher, I think, to put in a 2D.
That is a great suggestion. Thank you, Frank.
Mhm.
Yes.
Within the study, what's the significance or motivation for comparing the high-fat diet and the normal fat diet?
So that is the case of if you have so it's suggested that with a high fat diet, which is similar to a Western diet, that should cause results in changes in the gut microbiome. So we wanted to see that are the changes using B420, a probiotic, able to affect whether or not they had a bad gut microbiome or not. So with the normal fat diet, they should expect to have what's considered a relatively healthier gut microbiome compared to the high fat diet. And so if the B420 is able to significantly decrease in both situations, then anyone, regardless of diet, given B420 can have a reduction in their myocardial infarct.
Peter, what's the effect of high fat feeding and body weight?
Oh, there's an increase in body weight. So therefore, bad heart.
Increase in inflammation.
Yeah, and the inflammation. So the whole point of B420 was originally thought to be just sort of weight control only, compound. So that if we wanted to remove that uncouple and see whether or not is it due to the loss in weight, or is it due to the fact that it's actually just mitigating and becoming pro-inflammatory environment. So we do see a change in weight in high fat feeding, but yet with normal fat, you don't see a significant drop in weight. To effectively uncouple the weight effect, the drop in weight from the infarct from the actual compound itself.
Oh.
Yeah, so in your future directions, you mentioned adding estrogen to
Mhm.
So I was curious, is there anything in the literature that you've seen in humans on how hormone replacement therapy affects ileum and microbiome?
So I have not. So I will look into that because that is actually a very good question. We actually also considered possible controls doing a estrogen replacement model with the mice as well to see how that does affect the gut microbiome as well as the gut epithelium. So I definitely have to look into the literature for that. The only thing in regards in terms of hormone replacement therapy that I've looked into was the conflicting results of whether it has an effect on cardiovascular health. And so that kind of relates to the timing hypothesis where giving that hormone estrogen replacement therapy during that perimenopausal period is more beneficial for cardiovascular health than compared to once they already reached menopause. Adrian.
So this infarct sizing is not data of paper. Do you have infarct sizing? That's further down, isn't it?
What?
Do you have infarct sizing from your own data? Did you ever get infarct sizing or you had that stage?
No.
So this was all taken from somebody else's.
Yes.
Okay, just make-
Yeah. So this was preliminary work that was already done by our lab and published.
This was done by your guys' lab?
Yeah.
How come you wouldn't use the full life cycle? How come you exclude regions on your infarct sizing? The AHA 17 segment will encompass the entire section as well. So I think you should include that as well when you do infarct sizing here.
Are you referring to like the right part of the panel? So that's just showing an example of how we would measure the infarct size from one slice, but we would do it for all of the slices that are shown to the left.
But it doesn't encompass the entire ventricle. I feel like they're still missing. It would be a complete circle going all the way around. So
Also the supplemental.
It's
You can't
base-
see the
it's
right
base to
ventricles.
base.
okay.
So you slice them very thin, so you can actually get both sides. So effectively, you create a three-dimensional picture of the entire heart. And then, the ratios are determined from each of those slices. Up and down. That makes sense.
I had a general question about the effect of menopause on risk for cardiac events.
Mhm.
So I know you're talking about the risk or the severity of cardiac event increases, but is there an increase in risk for having one in the first place?
Yes.
Sorry, what was the last part? If after menopause, there's an increase in risk for just having a cardiac event.
Yeah. But the important thing that that you said was the timing hypothesis, right? It's not just in perimenopause. If you give estrogen back early, like the first 10 years of menopause, you protected. That terrible study gave it back to women in 76 years old who had seen estrogen for 25 years, and they were the ones who struggled. Right, and it was stopped because of breast cancer, not because of.
So Catherine might have had a question, otherwise we can, we're out of time. It's a little bit past noon, but Catherine has a quick question on Zoom.